Good morning students. This is 10th standard science biology video class video 1. In this video we are going to see the topic plant breeding and biotechnology. Dear students, this is unit 20 breeding and biotechnology. In this lesson we have already studied different types of plant breeding, animal breeding, structure of DNA, and also different types of newly formed plant breeding method. Today we are going to see the topic DNA fingerprinting and also genetically modified organism. Dear students, this is DNA structure and also DNA modification. Human genome has 3 billion base pair, that means nitrogenous bases. DNA is the important hereditary material which is present in the human beings, but the characteristic feature of identical twins is similar to all the characters, but they are showing variation in base pairs, that means nitrogenous bases such as adenine, guanine, cytosine and thymine. So that we are going to see the variation in the nitrogenous bases by the method is called DNA fingerprinting. This was developed by Alec Jeffrey. Each DNA showing the variation, that each variation and characteristic feature of an individual organism is identified by the method is called DNA technique. Here this is also identified by the sequence which is present in the DNA is called variable number of tandem repeat sequences. In human beings, 99% of DNA base sequence are the same. They are called bulk genomic DNA. Out of 100, only 1% of DNA sequence, that is nitrogenous bases, which is showing the variation or differentiation from one individual to another individual. That 1% of DNA variation is showing in the form of stretch-like repeated sequence. That means the nitrogenous bases are showing the repeated ones. That is called a satellite DNA. This is the important 2-mark question. That is 1% DNA sequence is present in a form of small stretch of repeated sequence is called a satellite DNA. So these repeated sequences are taken place in the form of number of copies is identified by the molecular marker is called a VNTRS, that is variable number of tandem repeat sequences. That should be identify the variation or differentiation of one individual to the another, and also finally the variation in the size of the DNA sequence or a segments is also identified by the method is called VNTRS. See the diagram, they are showing person 1, person 2, person 3. So these are all the base pair sequences. G for guanine, C for cytosine, A for adenine, T for thymine. So these are all the nitrogenous base pair which is present in the DNA sequences. That should be showing the variation in the 3 different persons. That variations are identified by the method is called a VNTRS method. That means variable number tandem repeated sequences. So this illustration is showing AGCT. That means adenine, guanine, cytosine and thymine is repeated 6 times in the first person. See the diagram AGCT. Varela is there. It is marked as in the blue color pointer. That is repeated in 6 times of the different person. That means first person. 5 times it is repeated in the second person. And also 7 times it is repeated in the third person. So this differentiation only identified by the method is called DNA fingerprinting. And also the DNA segment which is present in the third person is larger, because how many repeated sequences? 7 times is there, isn't it? Followed by DNA segments of first person which are having the repeated 5 times repeated person. Then the second person they are having the 3 times repeated person. So out of these 3, the third person only showing the 7 times repeated DNA sequences. So this is said to be the satellite DNA. These variations are identified in a group of population. நிறைய பேர் இருக்காங்கன்னா அவங்க கிட்ட இருக்கக்கூடிய வேரியேஷனை ஐடென்டிஃபை பண்ணுறதுக்கு they are using the method is called DNA fingerprinting. Now they are given the applications of fingerprinting. That means advantage of the DNA fingerprinting. So DNA fingerprinting techniques is used in the forensic application. What is forensic application? When you are seeing the cinema or a film, some murder is taken place in some other region, or a house means a person which are spreading a powdery like substance in the wall or a floor, table, floor wash, window, like that, to identify the fingerprints of the culprit. Those who have done the theft or a crime is called a forensic application. In that forensic application, they are using the technique is called DNA fingerprinting. Another one, they are also using to identify the parental character, father or a mother. That is also identified by the DNA fingerprinting method. Then second one, it also helps to study the genetic variation of the population. So we have seen the diagram, that is the 3% which are showing different DNA sequences. Likewise, we are going to see the genetic variation is also in the group of population. Then evolutionary sequence is also we can able to identify, that means parampariyam. And also speciation, that means how newly formed species which are produced is also identified by fingerprinting. So these are all the advantages of DNA fingerprinting. Dear students, now we move on to the last topic in this lesson is genetically modified organism. Shortly, we can able to name that GMOS. So this is the one of the method to produce genetically modified organism. The plants and animals is also produced by the transfer of gene from one plant to another plant, one organism to another organism, to change the modification of structure in the DNA is called gene transmission or genetic modification or genetically modified organism. So the organism which are produced by this method which are showing the desirable character, that means they are disease resistant character, noye taakat, and also pest resistant plant, poochi edume taakat, insect resistant plants is also produced by the method is called a genetically modified organism. So these GMO, genetically modified organism, which are showing the newly formed DNA, that technique is called recombinant DNA technique. See, which produce desired character, abdina special character. So this DNA fragment inserted, that means modified DNA by using GMO method, is transferred to the other organism. That gene is called a transgene. That means a plant or an animal which are having the defective gene, ஏதாவது false, ஏதாவது நோயை தாக்கக்கூடிய gene இருக்கு அப்படின்னா, that should be replaced by another correct gene or corrective gene by the method is called gene modification. So they are given the definition: plants are an animal which is expressing a modified endogenous gene or foreign gene is known as transgenic organism. So these transgenic organism which are showing the advancement or advantage, see, they are increasing the nutritional quality. And the சாப்பிடக்கூடிய சாப்பாடு பொருள் இருக்கு இல்லையா, we are getting from plants and animals only food material. Those nutritive value are quality. I also told resistant to disease. Any disease does not affected by the plants which are showing the transgenic gene. And also tolerant, that means they are facing any kind of environmental condition. Then by using this GMO, genetically modified organism, we can able to produce plant protein as well as the animal protein. Those proteins are used to prepare for medicines. That is said to be medicinally important, low cost, and also improve livestock quality. That means we can able to produce a medicinal or medicine from the plants and animals with low cost, and also we can improve the livestock quality. Livestock means panna, கோழி பண்ண வைக்கிறோம், மாட்டு பண்ண வைக்கிறோம், ஆட்டு பண்ண வைக்கிறோம்னு சொல்கிறோம் இல்லையா, that quality should be improved by GMO. Some genetically modified organisms are given here in the table column. First one, improved nutritional quality in rice. Gene inserted, that gene is called beta carotene gene. This gene beta carotene is present in the plant, which increase the vitamin A in human beings, because we are eating those plant parts. Those plant parts which are containing the vitamin A, this vitamin A increased in the human being by the injecting of gene is called beta carotene gene in rice plant. Another one, achievement. So by inserting this gene, we are getting the newly formed rice or genetically modified rice is called golden rice. This golden rice which produce beta carotene that can prevent the vitamin A deficiency diseases. Next one, increased crop protection, அதிகமான அளவுக்கு விளைச்சலை கொடுக்கும். BT gene, that means beta toxin gene from bacteria is called Bacillus thuringiensis. This Bacillus thuringiensis bacterial gene which removes the toxic substance against insects. ஏதாவது பூச்சி போய் தாக்கி ஏதாவது நோயை உண்டாக்குச்சுன்னு வச்சுக்கோ, that toxins are removed by the gene is called BT gene. Advantage, insects resistant plant, பூச்சியே தாக்காத plant நம்ம என்ன பண்ணலாம், உருவாக்கலாம். So these plants can produce the toxic protein that kill the insects with attack them. ஏதாவது இன்செக்ட்ஸ் வந்து அதை தாக்க வந்துச்சு அப்படின்னாக்கா அந்த ஜீனை நம்ம பிளான்ட்டுக்குள்ளே செலுத்திருக்கோம் இல்லையா, that plant should kill the toxic substance of insects. So this is the genetically modified plants. Another one, genetically modified animals is also produced. First one, improved wool quality and production. தரமான கம்பளி நம்ம என்ன பண்ணலாம், செம்மறி ஆட்டிலிருந்து தயாரிக்கலாம். Gene for synthesis of amino acid and cysteine. So when you are introducing this gene, the gene which are producing high amount of amino acid and cysteine. So transgenic sheep, அதாவது புதுசாக ஜீன் ட்ரான்ஸ்ஃபர் பண்ண செம்மறி ஆடை நம்ம என்ன பண்ணலாம், உருவாக்கலாம். ஸோ தீஸ் ஜீன்ஸ் ஆர் ஷோயிங் த எக்ஸ்பிரசிவ் கேரக்டர் வித் குவாலிட்டி டைப் ஆஃப் உல் கம்பளி நமக்கு கிடைக்குது. தென் இன்க்ரீஸ்டு க்ரோத் இன் ஃபிஷ்ஷஸ் ஆல்சோ வி கேன் ஏபிள் டு ப்ரொடியூஸ்ட் பை GMO. சல்மான் அண்டு ரெயின்போ ட்ரட் ஆர் திலேபியா growth hormone genes. That means these three type of fish, salmon or rainbow trout or tilapia, they are having the hormonal increased gene. Achievement is transgenic fish. The gene is also expressed good quality of fish variety with large size with contain large amount of protein. So dear students, these are all the different genetically modified plants and animals. So after this, dear students, after this we are going to conclude my video. Remaining we will see in the next video class. God bless you. All the best children.